Yeah, so I think all of us are familiar with the two main types of CAR T cells that are available on the market now, which either have a CD28 co-stimulated domain or a 4-1BB co-stimulated domain. Next slide. And the main manufacturing process of autologous CAR T cells relies on cells being collected from patients through a standard leukapheresis procedure. The cells are then undergo a period of activation and cell expansion, where the receptor is introduced and the CAR transduced cells finally expand over a 2-week period. And these cells are then available for infusion back into the patient. Next slide. If you look at where the field has moved in autologous CAR T cells, we've moved from the very first generation CAR T cells in the 1990s to the second generation CAR T cells in the 2000s. And it was only when the second generation receptors were introduced did we find that these cells were actually effective in patients. And the early studies in 2007 relied on CAR T cells being infused without lymphodepletion. But it was finally when lymphodepletion was introduced as a strategy before CAR T cell infusion did we see the results that we see today. And in 2017, we had the first approval for BALL, followed not long after by CAR T cell approvals for diffuse large B-cell lymphoma. And then not so long ago, we've had approval for mantle cell lymphoma and a further product, liso-cel, for diffuse large B-cell lymphoma. And I think we're all waiting eagerly this year for potentially CAR T cells to be approved for multiple myeloma. And there's a very good chance that that might happen. Next slide. But I think whilst undoubtedly the autologous CAR T cells have given us some very good results with about 50 to 60% remission rates in BALL, about 40% remission rates in diffuse large B-cell lymphoma, in multiple myeloma, we're not seeing long-term durable responses. Having said that, we are seeing very impressive early response rates. So I think there is still the prospect for improvement on these early products and these early trials. Next slide. So I think it's the case that with autologous CAR T cells, there are a number of challenges. One of the challenges is manufacturing failure, which can occur in lymphopenic patients. And the whole logistics of generating autologous CAR T cells is a significant undertaking. There is difficulty in treating patients with rapidly progressive disease, where unfortunately there just isn't enough time to manufacture the cells and get them back to the patient prior to the patient just deteriorating. And inevitably there is variation in the quality of the product from one patient to the next. And there are some disease groups, like for instance in CLL, where the T cells are fundamentally dysfunctional, and even when you introduce a CAR into them, the final product does not work so effectively. And all of this results in a very, very expensive product. We know that the list price for a CAR T cell product is something like $300,000 to $400,000. Now, I'm sure that will come down with time, but currently that is a significant limitation for many countries around the world. Next slide. So it's in order to try and address these challenges that allogeneic CAR T cells have been explored by the whole field. And when we talk about allogeneic CAR T cells, I'm not talking about CAR T cells derived from the donors following allotransplant. We're talking about off-the-shelf products now. So these are products which are pre-made. You have standardized, validated batches of cells that are only released when you know that they actually work. And because you can scale this up, you have reduced production costs. There is the potential for multiple infusions, something that is rarely possible in the autologous setting. And because these are cells that are pre-made from young, healthy donors, where you can pre-select a healthy T cell, it has the potential to be more efficacious than patient-derived autologous CARs. Next slide. But what are the obstacles to allogeneic CAR T cells? Well, the biggest obstacle is the fact that you've got a HLA mismatch product here with the potential to cause graft versus host disease mediated by the donor TCR. These are cells that can be rejected by the host immune system, and the rejection can occur through a variety of different means by both T cells, NK cells, and B cells. We know that even in the autologous setting, you get infections as a potential complication. In the off-the-shelf CAR T cell field, there is the prospect of even more severe infections, partly because of the more intensive lymphodepletion that is generally used for this product. Next slide. So what are the strategies that we can undertake to try and generate an allogeneic CAR that can be given to any patients? Well, in this schematic diagram is shown the barriers to this and the strategies to overcome these barriers. So in most cases, you'll see that the endogenous T cell receptor needs to be disrupted so that this CAR T cell that's introduced into the cell can actually proliferate without causing graft-versus-host disease. And this is generally done by a variety of different gene editing techniques to knock out the TCR alpha gene. There is also the prospect of knocking out the MHC class 1, and one way of doing that is to knock out beta-2 microglobulin. That's something that a number of different groups have tried to do. But what about the host immune system? You've got to find a way of preventing the host immune system from rejecting these cells. And one of the ways in which people have tried to do this is by using an intensive lymphodepletion, using an agent like alemtuzumab, an anti-CD52 monoclonal antibody. And this gives profound host immune lymphodepletion and prevents the CARs from being rejected. But in order to make the donor CAR T cells invisible to the alemtuzumab, because otherwise they would just be killed by the infused alemtuzumab that has a long half-life, the cells can have their CD52 knocked out. So now these cells can no longer be seen by the alemtuzumab and expand via the CAR. Next slide. So the very first off-the-shelf CAR T cell product that went into patients was this one, UCAR19. And I've been fortunate to work with this through one of the trials. And the strategy employed with this specific product was to use TALENs to edit out the TCR alpha gene and the CD52 genes. Lentiviral vector was used to introduce the CAR, which had a 4-1BB co-stimulated domain. And it also had a CD20 suicide gene built into it, which in theory allowed these cells to be eliminated by the use of rituximab. Next slide. The manufacturing process involved collection of cells through a standard apheresis procedure from a healthy donor. The cells were activated, lentivirally transduced to introduce the receptor. The 2 TALENs for 52 and for TRAC were electroporated into these cells. They went through an enrichment process then to select out for just the TCR alpha and beta negative cells, and then frozen down, ready to be infused into patients. And you'll see that these cells are 99% free of TCR alpha beta cells, although there is still about 1% of cells that still express a donor TCR, which have the capability of causing graft-versus-host disease. Next slide. So these are the 2 trials that have just been completed now, looking at UCAR19. First one was the CALM trial, and this was in adult BALL. It was designed as a phase 1 dose-finding study with 3 different dose cohorts. The pediatric study just had a single flat dose, but using the same product. Next slide. And the preliminary results from these 2 studies were recently published in the Lancet. And what you'll see from the baseline characteristics of the patients, at that time there were 14 patients treated on the adult study, 7 in the pediatric study. You'll see that the majority of the patients had had more than 4 prior lines of treatment. More than a third of patients had had either inotuzumab or blinatumomab, and the majority had had a prior allogeneic stem cell transplant, so a high-risk group of patients. If you look at the disease burden going into the trials, they ranged from MRD levels of disease to frank relapse. Next slide. So what about the toxicity profile that was seen in the study? Now, 91% of the patients developed cytokine release syndrome, but only 15% had grade 3 or more CRS. So I think this is in keeping with what you might expect to see in CD19 CARs for BALL, for instance. Neurotoxicity was seen in about a third of patients, but again, it was mainly grade 1. And very fortunately, we only saw 2 cases of graft versus host disease, both of which were grade 1. We did see a lot of infections. 62% had some form of infections, mainly grades 1 and 2, although we did see more viral infections than you might expect with the autologous CARs, and that is predominantly because of the use of alemtuzumab in the lymphodepleting regimen. We did see some cytopenias, and this is again a common finding amongst patients that receive CAR T-cell products. But patients that had prolonged cytopenia in our study was about a third of patients. Next slide. So this was the efficacy reported in those 21 patients treated. And do bear in mind that the way the study was designed was that the patients received a CAR infusion following FCA lymphodepletion. Once the patients achieved a remission, then the majority of them went on to have an allogeneic stem cell transplant. And the main rationale for this was because the expectation was that persistence of off-the-shelf CARs may be limited, and therefore one ought to take the opportunity to consolidate these patients with an allo stem cell transplant. And you'll see that 67% of the cohort as a whole achieved a complete remission following UCAR19. This was at day 28 post-treatment. That's 14 out of 21 patients treated. But 4 out of those 21 patients did not receive alemtuzumab. That was just because of investigators' choice of worry about viral infections. And you will see that none of those patients who did not receive alemtuzumab responded. So if you were simply to look at the cohort that had fludarabine, cyclophosphamide, and alemtuzumab, 82% of these patients had a complete response, suggesting that you do need alemtuzumab for expansion of this particular CAR T-cell product. You'll see in the triangles that there were a number of deaths that occurred following the transplant. So some would have been standard transplant-related complications. Others were deaths from progressive disease. Next slide. And I guess whilst the initial response rates were very encouraging, if you look at the durability of the response, this was rather disappointing, with a PFS of only about 27% at 6 months and overall survival of 55% at that stage. Do bear in mind, though, that for the adult study, half the patients would have had very low doses because this was a dose-finding study. And the first dose in the adult study was only about 1 × 10⁵ cells. So considering the cell doses were rather limited, an early response of 82% in alemtuzumab-treated patients, I think is an impressive first start for the field. I think the challenge is now to try and improve the durability of these responses. In the next slide, you'll see perhaps why the responses are not that durable. Next slide. So this slide shows the expansion kinetics of the CARs, and you'll see that although in the majority of patients who received alemtuzumab, you get an initial expansion of CARs as expected, peaking at about day 14, you then get a gradual decline by day 28, and beyond day 42, there were only 3 patients that had persistent CARs. And this fundamentally might be the reason why you're getting relapses occurring. Now, in not all patients could you assess the long-term kinetics because they all went on to have allotransplants, so you would ablate the CARs in any case. So I think it is still unclear as to what sort of long-term persistence you would see if, for instance, these patients did not have a subsequent allotransplant. But what was clear in the slide below, you'll see that expansion did matter. If you didn't expand, you didn't respond, shown down here. Next slide. So the field is now starting to look at other ways of generating allogeneic CARs. And a number of groups have looked at genome editing ways of generating this. And the way one way of doing this is to actually generate a CAR, a anti-CD3 CAR that actually functions to retain the CD3 complex within the endoplasmic reticulum and stops it from being expressed on the cell surface. So in other words, you're getting rid of the TCR, but not by using a gene editing technique. And this is another CAR that will go into the clinic hopefully very shortly. Next slide. The Memorial Sloan Kettering group have looked at ways of introducing, of disrupting the TRAC locus by introducing the CAR into the TRAC promoter area. So it achieves two things. It disrupts the TRAC locus, but it also ensures that the CAR is now under the constitutive expression control of the TCR promoters. And what they've shown, certainly in their preclinical work, is that you get very stable expression of the CAR in this context without any expression of the endogenous T-cell receptor. Next slide. So these are some of the ongoing allogeneic off-the-shelf CAR T-cell trials. Allo501 was a study reported in the US at ASH last year in DLBCL, again showing some encouraging early responses, but perhaps not durable responses. UCAR123 is a off-the-shelf CAR that's being explored in AML. Similarly, CAR against CS1 is also being looked at in myeloma. And the Allogene group have also reported their early findings of an anti-BCMA AlloCAR in multiple myeloma. Again, early encouraging responses, but the proof will come in whether or not you can make these responses durable. Next slide. So these are some of the alternative off-the-shelf cellular therapy products that are being used to introduce CARs into patients. NK cells have had a lot of publicity from the work done by Katie Rizvani at the MD Anderson. Gamma delta cells can also be used in isolation, but also by introducing a CAR into them. The invariant NKT cells are also being used, and NK cell line CARs and iPS-derived CARs. So I think you're going to see in the next couple of years a whole series of different types of off-the-shelf CARs that are being explored. Next slide. So just to summarize, allogeneic CAR off-the-shelf CARs can be an option now, where autologous CAR T cells are challenging or unable to be used. We demonstrate from the CARM and PALL trials that it is feasible and it is safe to use allogeneic CARs, but there are still some issues with toxicities and lack of persistence that we do need to overcome. And I think the field will move forwards with the next generation allo CAR T cells. I just want to end with a final slide, which just throws a number of questions. So these are some of the questions for the allogeneic CAR T cell field: whether or not they can persist long enough for durable disease control, who makes a good allogeneic CAR donor, and ultimately, will off-the-shelf allo CARs be cheaper and more accessible? And after all, that's the main purpose for this. Thank you.